All right, good morning. This is Rex Bernardo at the University of Minnesota. Thank you for joining my class today. I teach a class, Agronomy 8202, Breeding for Quantitative Traits in Plants. And this is the last lecture that I have for the semester. We have a capstone event next Thursday, breeding games competition, and that's it for a class. And I figured because I'm teaching this via Zoom anyway, I might as well open it to others who might be interested to hear about a possible reinvention of quantitative genetics for plant breeding. Some ideas that for us to think about, and I organized my thoughts according to this old English rhyme, something old, something new, something borrowed, something blue, that many a bride is familiar with. This session is going to be recorded, so for those who might be interested, if colleagues interested in the recording, let me know and I could forward the Zoom link later on. I'm thankful for that many of the thoughts that I'm going to express today were concocted over espressos while I was on a sabbatical leave at the University of Bologna in Italy. And I thank Professors Elisabetta Frascaroli and Roberto Tuberosa and their colleagues for their very warm and kind hospitality while my wife and I were there for part of last fall semester. Also, an article of the same title has been published in the current issue, it's online, open access, in the Heredity journal based in the UK. And this is part of a special issue that they're having. So many of the thoughts, or in fact, almost all of the thoughts that I'll be expressing today are already in that article that I invite you to view. However, I thought it also is, even though that has been published, that it is helpful to have a webinar such as this because it allows for a two-way dialogue as opposed to more of a one-way conversation that is typical of a journal article. So, reinventing quantitative genetics for plant breeding, something old, something new, something borrowed, and something blue. And why all this? To do that, it's useful to take a little bit of a history tour of quantitative genetics. And talking about history, let me talk a little bit of my own. I was a graduate student at the University of Illinois in the mid-1980s. And in spring 1986, I took a course under Professor Michael Grossman at the University of Illinois in the Animal Science Department entitled Quantitative Genetics. And that was my first exposure to the field of quantitative genetics. And at that time, I learned about this article by Ronald A. Fisher, Sir Ronald A. Fisher, called The Correlation Between Relatives on the Supposition of Mendelian Inheritance. And to me, the title of that article seemed a bit odd. Why the supposition of Mendelian inheritance, and why the correlation? Why focus on the correlation between relatives? Why not look into the mean, and why not look into the variance and focus on those as we were doing, or as I was expecting in the class, being an agricultural department in an ag campus. And it was only much later when I learned about the background to why Fisher wrote this article. And so this article, classic 1918 article, was in the aftermath of a very heated debate, very heated argument, I would even say, between two groups of English scientists, the Biometricians that were led by Carl Pearson and Raphael Weldon, and the Mendelists, on the other hand, who were led by William Bateson. And so the origins trace back to the school of biometry for which the main proponent, main scientist was Sir Francis Galton. And Galton was interested in the variation among human traits or characteristics, as well as the possible inheritance or the passing on of these characteristics or traits from parent to offspring. And so Galton was able to accumulate large data sets on different human characteristics, such as the length of the forearm, but mainly he focused on human height. And it was from these studies that he actually came up with this concept of regression toward the mean. Actually, he called it regression toward mediocrity. The main idea then is that if you have two parents who are much taller than average, then their offspring would be less tall than them. The height of the offspring would be regressed towards mediocrity, which meant towards the mean. On the other hand, if you have two parents who were shorter than the average of the population, the height of, on average, it would vary, of course, but the height of their offspring would be regressed back towards the mean. So you have that shrinkage toward the mean and this regression toward mediocrity. And from his observations, Galton estimated that for height in humans, the regression coefficient, if you would, toward the mean for height was 2/3, meaning that if a mother and the father exceeded the height of the population by 9 centimeters, it means that the height, the average height of their offspring would not be 9 centimeters, but it'll be 2/3 of that. And so it would exceed the height of the population by 6 instead of 2/3, instead of 2/3, instead of 9. And so Galton then, Pearson was Galton's protege, and Pearson further investigated the inheritance among humans, and in that process looked at the correlation between height and forearm length and so forth, correlation between different kinds of relatives in the human population, parent offspring, of course, and uncle, nephew, and cousins and so forth. Brother, sister was an important type of relationship. Now, around that time, or a bit later, in 1900, Mendel's experiments were rediscovered by Correns, De Vries, and von Chermak. And with the appearance then of Mendelian inheritance, the Mendelists thought that because they were looking now at discrete inheritance, that by definition, De Vries thought, by definition, any continuous variation by definition cannot be inherited, because Mendel had shown that it is discrete characteristics that were passed on from parent to offspring. And it was in the aftermath, towards the end of this heated debate, that Fisher's paper came up. And that's why it was on the supposition of Mendelian inheritance. And it also explains why he focused on the correlation between different types of relatives, because that was the subject matter of the debate between the biometricians and the Mendelists. And Fisher showed then that according to the basic model for quantitative genetics that we hold to up to today, that if we have a large number of loci and a random mating population, and these loci behave independently, that through the joint action of these multiple unknown loci, we could come up with the same correlations between relatives that were obtained empirically by the biometricians. And therefore, this served as a bridge between these two schools of thought, the biometricians and the Mendelists. And so in doing so, Fisher invoked several assumptions that we hold to even today with classical quantitative genetics. And I've mentioned two of them. One is random mating. A second is the independence of this inherited factors. And Fisher then further partitioned the genetic variation into that is passed on from parent to offspring, the genetic effects that are transmissible from parent to offspring, and the genetic effects that are not transmitted from parent to offspring. We now know that these transmitted effects we call today as the additive effects. The additive or the average effects of alleles are that which is transmitted from parent to offspring, whereas the non-additive portion is not transmitted because genotypes are formed anew after fertilization. In hindsight, instead of additive variants that we talk about today, it might have been more accurate to call this as transmissible variants or hereditary variants, because I think that really captures the essence of what Fisher was looking at, the effects that are transmitted from parent to offspring. So therefore, it's the hereditary variants. Fisher also then revisited the parent-offspring correlation for height in humans, and he found then that the regression isn't 2/3, exactly 2/3. I mean, it can be 2/3, but more precisely, it is a function of half of the additive variance, and it is half because it's the height of one parent, and then the other parent being assumed uncorrelated in terms of height to to that parent in question. Of course, Fisher also looked at assortative mating, but so that's another, it's a complication he looked at, but for the most part, he derived it as half of the additive variance, and that is what we see in plant breeding today. For example, with mass selection, without pollen control, meaning looking at just the effect of one parent on the offspring, indeed, the breeder's equation as derived by Lush in 1937 is a function of a half of what we now call as heritability. So we see here then that these very foundations that Fisher came up with relate very closely to the response to selection and other concepts in quantitative genetics that we have today. And so this ushered then, quantitative genetics ushered a lot of developments in the breeding for complex or quantitative traits in plants. It started, for example, if you go back to the 1940s, there were a lot of investigations on the genetic basis of heterosis. The 1950s and the 1960s have been called as the golden era of quantitative genetics and plant breeding, because in these 2 decades, we see a lot of investigations on genetic variances, mating designs to estimate genetic variances, empirical estimates of variances in different populations. We see work on genotype by environment interaction. We see work on stability analysis. We see work on index selection, really a lot of the foundational concepts that we use to this date. In the 1970s, we saw then the use of isozymes in plant breeding. We saw, and then the 1980s began some formal work. My advisor started to do some formal and I think long overdue work on formal methods for choosing parental germplasm in a breeding program. And so these contributions through quantitative genetics continue on today. However, if we pause, we can easily see that plant breeding today is markedly different from the plant breeding the way it was in 1918 or 1920 or 1930s or 1940s, or even 1980s and 1990s. Plant breeding today has changed a lot. And so I think there are 3 main reasons why we think we might consider reinventing quantitative genetics for plant breeding. And these 3 reasons are, first is that the expectations of plant breeders from quantitative genetics have changed. Second, the key assumptions in quantitative genetics are unmet in modern plant breeding programs. And thirdly, we have new tools that are available to us today that were not available to Fisher in 1918 or to those who followed, who had major contributions to quantitative genetics and plant breeding in the decades that followed later on. And so, in 1969, my advisor, John Dudley at Illinois and Moll, published an article on the use of genetic variances in plant breeding and in general contributions of quantitative genetics to plant breeding. And in this article, they raised several questions that could be answered by quantitative genetics, but these several questions could really be distilled into 3. And these 3 questions are, which germplasm should I use? Second, what type of cultivar or variety should I develop? And third, what breeding methods should I use? Which germplasm? What type of variety? A pure line, a synthetic, an open pollinated, or a hybrid variety, single cross, double cross, and so forth. And third, what breeding method should I be using? And so, for example, if we think of what germplasm, we can say that, well, the good germplasm in plant breeding would combine a high mean performance for several traits, as well as a large genetic variance for these traits of interest. And then the combination of the mean and the variance, in fact, has been formalized into Schnell's usefulness criterion that looks at the mean of a population, the mean of the superior individuals, say the top 10% in the population. So therefore it combines mean and genetic variance. We can look at different types of genetic variances to see, for example, what type of variety might be useful. For example, if you have a trait such as maize grain yield, for which there is a lot of dominance variance, then that would tell us that hybrid varieties would be useful because because heterosis is mainly due to dominance, and therefore dominance leads to dominance variance, and therefore if there is dominance variance, we could grow hybrid varieties to exploit that heterosis. In hindsight, though, it doesn't take quantitative genetics to answer these questions. That breeders can choose good germplasm pretty much just by looking at the mean performance. And in this day and age, we can analyze different germplasm with molecular markers to see clusters of similar and dissimilar germplasm. And so therefore, we do not really need any formal quantitative genetic investigations, or so it seems, to identify what might be the best germplasm available. As far as the types of varieties, these can be answered, a lot of it by mode of pollination and by whether simply the parent, a hybrid, is superior to the inbred parents. And so if a hybrid is superior to its parents and hybrid seeds can be produced feasibly, then we can have hybrid varieties. Otherwise, we look at other types of cultivars, be it a pure line or a multi-line or a synthetic cultivar. Now, the third question has been quite interesting to think of. What type of selection procedures should be used? When I was in school, I was so ingrained into different recurrent selection procedures. In fact, I remember my professor in plant breeding, I think it was Agronomy 323 at Illinois at that time. My professor in plant breeding was a maize breeder and geneticist, and we discussed at length different recurrent selection procedures, half-SIB recurrent selection half-SIB recurrent selection, or full SIB, or S1 recurrent selection, or reciprocal recurrent selection, or recurrent selection with an inbred tester, and so forth. When I got my degree and started working with a private company with Limagrain in 1988, I found they did not use recurrent selection at all. It was pretty much a straight pedigree breeding program where we crossed two parents, developed new lines, tested them for their hybrid performance, selected the best lines, and repeated that same procedure more on a line development basis. So I think there was an era, in fact, where these questions were very meaningful, because there were concerns early on that pedigree breeding would very quickly exhaust genetic variation, or it would not allow enough recombination to get the best recombinants from a cross. But just from empirical work, we've found that if we have a wide enough germplasm base to start with, and we do pedigree breeding repeatedly and introduce new germplasm every once in a while, that we can indeed sustain this progress. And so I think in this day, the question of which breeding method to use has actually become quite different, and it has become more of a factory type of process that I'll talk about in a little bit, but that classical quantitative genetic machinery isn't quite well equipped to answer. The second reason that I mentioned is that the assumptions are unmet. So Fisher made these two key assumptions. One is a random mating population, and second is the lack of linkage, or more precisely, the assumption of linkage equilibrium. Now, if you're doing recurrent selection, you could very well argue correctly that you have a random mating population, because each cycle of selection is the product of random mating, and so therefore that is a random mating population. But now, if you look at most breeding programs as a whole these days, the breeding germplasm as a whole does not constitute a random mating population, nor does it really constitute pockets of random mating populations. Instead, you have distinct, diverse lines of different geographical and genetic origins that the breeder has found useful. And these distinct lines are not random mated, but they are paired in specific combinations to come up with new progeny. So really, if you think about it, this idea of random mating does not really apply to contemporary plant breeding as we see it today. The second reason, it's interesting because when I was a grad student, I was doing my thesis work was relying actually on the assumption of linkage equilibrium. And the population I was working with was random mated 2 times in order to come up with more linkage equilibrium. And when I was a student, we thought of linkage as the plague. We did not want linkage. We wanted to avoid linkage at all costs. And in fact, if I'm honest about it, I was closing my eyes and saying, well, I'm just assuming that linkage is either absent or is weak, that I don't have to worry too much about it. And it's interesting that with the advent of molecular markers, suddenly linkage, this equilibrium became a hero. We wanted it. Instead of saying we don't want linkage, now we want linkage disequilibrium because we're relying on that now to find marker trait associations. And so the point being that regardless of whether linkage is a villain or not, the fact being is that linkage is with us, and the classic assumption that Fisher had to make, because there was no other recourse. There was no other recourse for Fisher. He had to assume random mating and linkage equilibrium. Those do not apply in reality. But at the same time, quite wonderfully, we now have tools so that we do not have to make those assumptions anymore. And so that is the third reason for trying to reinvent quantitative genetics for plant breeding, is that we now have tools to radically change the way we look at quantitative genetics. And so these two main tools in my mind, the first one I've talked about, the first one is molecular markers that, as I mentioned, began as isozymes. We were investigators looking at a dozen isozymes, maybe up to 20 isozymes at a time. And then these became more numerous with the development of RFLPs, or restriction fragment length polymorphisms, in the 1980s. And then with simple sequence repeats, and finally with SNP markers, we have plenty of molecular markers that we can use these days. And so the wonderful thing is that instead of having to estimate or to assume random mating, we can actually track the inheritance of chromosomes from parent to offspring. We can track how linkage, how crossing over breaks up the chromosomes, chromosome configurations present in the parent, and how they're passed on to the offspring, and how segments of chromosomes are shared between relatives, meaning that we don't have to assume random mating anymore, but we actually have the means to see the segments, a mosaic, if you would, of the genetics that is underlying the inheritance of the organisms that we're studying. So I'm saying we're not quite at the gene level there, but we're so much closer than Fisher was into looking at the inheritance now of segments of chromosomes, instead of just trying to look at probabilistic models. The second tool that has become available is fast and big computers. I remember one of my committee members in my PhD defense was Professor George F. Sprague. He had a long time distinguished career as a maize breeder, and he was the one who came up with the classic concepts of general and specific combining ability in crops. And Professor Sprague told me that in his day, he could not have winter nurseries. And the reason he could not have winter nurseries is that it took him and his colleagues a long, long time to analyze the fall data with the computing machines that they had. And the data could not be analyzed in time to make decisions as to what to send to a winter nursery. And so therefore they just decided we're not going to have a winter nursery because we can't crank through the data quickly enough. When I was a scientist with Limagrain until 1997, the fastest computer that I had back then was a Pentium 90 machine with 64 meg of RAM. 64 meg, not gigabytes, 64 meg of RAM. And I remember buying that computer via the phone, and I told the person I'd like a machine with 64 meg of RAM, and there was silence on the other end. And finally, the sales lady asked me, what do you need that for? And I had to explain, well, I'm doing a lot of computationally intensive work. And with 64 meg of RAM, I remember I had to be quite stingy in the programming that I was doing, working with large matrices for specific combining ability effects. For example, the matrices were large for SCA, but it was sparse, a fairly sparse matrix in which many of the elements were zero. So I would have this matrix, but before I could proceed to other calculations, I had to free up the space occupied by that large matrix. And the way I did it was I stored the elements of that matrix in three columns. First is what is the row, what is the column, and then the non-zero element. meaning that if there was no such row and column index, then it was a 0 element. So I stored it in that scratch pad, if you would, before I could go to something else. And now we're at a time when that really is not an issue anymore, and we can do a lot of fast and big computations, as many folks in our field have done. So these tools have radically changed the way what we can do and how we can do quantitative genetics and plant breeding. So with that as a backdrop, I thought it's helpful then for us to think about what might a new quantitative genetics for plant breeding look like. What are some things that we need to hold on to? Or what are some things we might need to introduce, and what are some things we might need to borrow from other fields for quantitative genetics to be as useful as possible in breeding traits of breeding crops for different quantitative traits. And so 2 caveats. One is what I'm going to propose is applicable mainly to plants and improving plants for quantitative traits. They might not apply to other fields such as human genetics or animal improvement, for which quantitative genetics has also played a key role. Second is that one may question whether such a reinvention is necessary. Or on the other hand, we could just let the field evolve like a language. If we think of the language we use, the words we use, a language naturally evolves until new uses of different terms become accepted in common day practice. And you may argue, and one might argue rightfully so, that, well, it doesn't really matter if, as long as we're using that same terminology, if everybody is using the terminology and approaches and methods in the same way as others, then we can still effectively communicate with each other. Now, my opinion is a bit different. My opinion is that as scientists, we want to be precise. If we want to be precise in our science as scientists, then we also need to be precise in the language that we use. And so therefore, my opinion, and feel free to disagree, my opinion is that it is better to carefully think of new concepts that we might have to introduce to accommodate new things, rather than use old concepts to accommodate new developments that those old concepts were never designed to accommodate in the first place. So with that as a lengthy introduction, I would like to propose 8 ideas again, arranged according to this British rhyme of something old, something new, something borrowed, and something blue. And I've got visual aids here. First one, old, P equals G by E. That does not change. We're looking at phenotypic values, that which we see, that which we evaluate in the field, in the nursery, in the greenhouse, and we will continue to say that this phenotypic value is due to the joint effects of genes that are passed on from parent to offspring and that are shared among relatives, plus some residual non-genetic effect. Of course, there are ways to further partition G into different components if we wish, and there are different ways to partition the E component, but this basic notion still stands: P equals G plus E. That doesn't say anything about the underlying G, but it does still say that age-old equation still holds. Second, genotypic value, the G component, will continue to be the component that we as breeders focus on. We are interested this day in finding, the main goal then of a breeder today is to find those candidates, those clones, those lines, those individual plants, those test crosses, those hybrids that have the highest G. And we now have several ways to do this. One is going back to the P equals G by E. We can assess G according to the phenotypic value of that individual when it is evaluated in the appropriate environment. But there are other ways by which we can evaluate G, and that has come to the fore recently with more prediction-based processes, such as genome-wide prediction. That with genome-wide markers, we still need some baseline P phenotypic data from the training population, but using those phenotypic data and molecular markers, we can use prediction-based approaches to evaluate the G component without having to phenotype these very individuals that we are interested in. And it does raise the question, what then becomes our gold standard? What becomes our gold standard for evaluating G? Right now, our gold standard for evaluating G, I think, at least in my mind or in our minds, remains P. That when we try to look at the usefulness of genome-wide predictions, we always compare it to phenotypic values, with the implicit assumption that the P is, so to speak, our gold standard. That's what we look at. And I think with more and more phenotypic data in training populations and with genome-wide predictions being more prevalent, we need to seriously start thinking, should that P of an individual remain the gold standard, or should G from genome-wide predictions start to become our gold standard? In other words, can it be possible? Would it be possible? And if so, when would that happen? That the predictions of genotypic value are actually more accurate and more precise than phenotyping that we would do, that those practitioners who are used to phenotyping know that those data could be noisy anyway to start with. Something to keep in mind. Another old thing is that major QTL will continue to be important. In the past few decades, there has been a lot of work on QTL mapping in plants, and these have yielded good results as far as finding major QTL for abiotic and biotic stress resistance, and these QTL then have been deployed widely in breeding programs. And then this effort in finding major QTL should continue. It's now wonderful that in a way that this has become routine. As we look at major QTL, I think it is important to define what a major QTL is. And it's interesting that it really is not, it's a bit difficult to come by a formal definition of what is a major QTL. And my definition of a major QTL is one that has an effect that is large enough and consistent enough to be useful in a breeding program. One that has an effect large enough and consistent enough to be useful in a breeding program. And you may say, Rex, that's a subjective definition, and it absolutely is. Because what could be a major QTL to one breeder in one breeding program might not be a major QTL to somebody else. And to define a major QTL, we should then look at the effect of the QTL on the mean for that trait rather than the R-squared. To illustrate, in the past, we've done work at the University of Minnesota on some high oil corn germplasm. And we've found that a candidate gene, DGAT, has an R-squared of 27% for kernel oil in maize. And you may say, and we would say, yes, R-squared is a wonderful R-squared, it's a high percentage. But if we looked at the effect on the trait, it would increase oil percentage by 2 percentage points, meaning that if oil in maize is 4%, by introducing 2 copies of this allele, now instead of 4%, it becomes 6%. From that perspective, I concluded, well, it really is not a major QTL because we can have cultivars that are present that have a much higher oil percentage, 8, 9, 10%. So again, I think we need to be careful in defining QTL, not according to the R-squared value, because it's a bit difficult to interpret from a breeding standpoint, but really what is the predicted change in the trait when we introduce this major QTL into our germplasm. Now, looking at what might be new. First new thing that I'd like for us to think about is that we shouldn't have to assume Hardy-Weinberg equilibrium anymore. It is a nice idealized concept, but in plant breeding, unless we are working with recurrent selection populations, then we don't have a Hardy-Weinberg equilibrium. The other exception is that if we are working with the F2, the F2 of a cross between 2 lines, then this would indeed be in Hardy-Weinberg equilibrium at one locus, because the genotypic ratios 1:2:1 at segregating loci are what we would expect at Hardy-Weinberg equilibrium. Not assuming Hardy-Weinberg equilibrium also means that we don't need a reference population anymore. To illustrate, for example, if I were a breeder, I like that term, and I'm crossing two lines, and I get and I get progeny from that line, and then I develop and I look at the mean and the genetic variance in that population. By the time I get the estimates and the mean and genetic variance, I don't really need to estimate back or make inferences about the base population. From a breeding standpoint, I'll simply select the best lines without having to make inferences to the base population from which those lines are derived. In other words, from a practical breeding standpoint, there really is no need to have that reference base population if our main interest is finding the best individuals in a segregating cross. Secondly, this idea of not having to assume Hardy-Weinberg equilibrium also brings to the fore a divergence between two schools in quantitative genetics: the Edinburgh, Alan Robertson, Douglas Falconer school that looked at arbitrary allele frequencies in random mating populations, and the Mather and Jinks school that looked at crosses between lines and allele frequencies of half. And in a way, this whole notion of not having to assume a random mating population brings these two together in the sense that, well, if we're simply looking at a cross between two lines, then we can take a look at the Mather-Jinks approach and also translate that in terms that are familiar to those in the Edinburgh-Falconer school. Now, if we say then that we don't need a Hardy-Weinberg equilibrium, it also would indicate that we do not need to define anymore additive variance, dominance variance, and epistatic variance. So to the students in my class, I'm sorry that we talked a whole lot about additive and dominance and epistatic variance. And here I'm saying we don't need that anymore. Well, actually, we still need that, at least for a communication standpoint, because these concepts will continue to be useful in other fields as well as in the classical literature. Now, why do I say we don't need these concepts of additive variance and dominance variance and epistatic variance? For one thing, plant breeders, are not really as interested in breeding value as they are in the value of that individual itself. Let me say that again and I'll explain it. Plant breeders really are not interested in breeding value. They're interested in the value of individuals itself. It's a bit different from animal breeding. For example, if you take a dairy bull and you take the milk yield of a dairy bull, which is 0, but the dairy bull has genes for milk yield that it can pass on to its female progeny, then the breeding value of that dairy bull is of utmost importance because, again, the farmer will not be using that dairy bull to produce milk, but it's that dairy bull's progeny. It's quite different, for example, if you're a cassava breeder, you're interested in the performance of that cassava clone itself. You're much less interested in the performance of its progeny. You're primarily interested in that cassava clone because that is what you'll be growing. And so we see then that the ability to replicate plants is quite different from animals. And I see a comment here says that's not true for poultry and swine breeding, at least. Thank you for that comment. And so, but the point is, and just focusing on plants, we're interested mainly not in breeding value, but in the value of that candidate itself. Now, the additive variance is the variance due to breeding values. And so therefore, if we say that breeding value really is not of much interest, and additive variance is the variance of breeding values, then we are not as interested then in breeding value. And also, breeding value is defined according to this assumption of a random mating population, which we said does not hold true anyway for the most part. And so therefore, it seems time then to not worry too much about additive variance. And if we don't worry about additive variance, then we shouldn't worry too much about dominance and epistatic variance because those were derived from the same framework as additive variance. And so you may ask then, if we do not estimate these types of variances, what do we estimate then? Well, the easy answer is we estimate the variance due to the candidates. meaning that if we're looking at recombinant inbreds, then we estimate the variance among recombinant inbreds. If we're looking at single cross hybrids, we estimate the variance among single cross hybrids, and we can further break that down to general and specific combining ability variance. If we're looking at clones, we estimate the variance of clones. And so again, it's a simple framework in which we forget the underlying genetic interpretation of these variances according to Fisher model, but we stop at these empirical estimates of these variances among the candidates we have and say that is good enough for the purposes that we have. Now, then if we say we're getting rid of a random mating population, we're getting rid of additive variance, it also means that we need to get rid of heritability. When I say get rid, I'm being loose there, that we don't focus on heritability anymore. So when we talk of concepts now that we need to borrow, I think that we need to borrow concepts of reliability and least significant difference, which is quite familiar to us as breeders to start with. Let me explain that. Let me explain that a little bit. So we have narrow sense heritability, which is due to the additive variance. And then we have broad sense heritability, which is additive and non-additive. And in a way, broad sense heritability is an oxymoron because it measures variance due to things that are not heritable, not passed on from parent to offspring. And it seems that in plants, at least way back to the 1960s, the concept of heritability in plants has been muddled quite a bit. And a reason for that is if you look back to Fisher's work, it was measurements on individual individual human beings. Whereas when we look at plants, we look at half-sib family, we can look at recombinant inbred lines that are replicated to different levels in a single location or multiple locations. So we have measurements on different numbers of individuals. And the interpretation of heritability then depends on the unit of measure. Are we measuring the mean of several individuals or just one individual? Are we looking at mean across several locations? Are we looking at it at the plot level? And so it seems to me that heritability has been used mainly as a measure of the importance of nature versus nurture. How important are genetic effects versus non-genetic effects, nature versus nurture? And if that is the case, then I think it might be simpler if we borrow a concept from other fields called reliability. Reliability is a concept that has been widely used in testing, in education, in the industry, and in other fields. For example, a university admissions test is reliable if, for example, the same student taking different versions or different iterations of that test will have constant scores, assuming, of course, that the test taking, that the ability of that student does not change between repeats. So if an ACT test is reliable, then that same student would take the test and might score a 28 on one, 27 on the next test, 28 on the third, and so forth. If it's all over the place, then we say then that test is not reliable. And you see the easy transposition of this concept to the varietal trials we have. We say that measurements of the performance of a candidate we have in a breeding program are reliable if they stay fairly constant across different environments. They're not reliable if they swing from one end to the other end. And reliability, it turns out, one way to calculate reliability is it's exactly the same way as we calculate heritability. And that would be then, it's equal to the variance of the subjects divided by the variance of the subjects plus random error. So it's an intra-class correlation, which is very similar to heritability. So in a way, it's having this new concept that respects the definition and derivation of heritability as has been done, yet it is a new concept that is based on clear definitions and clear assumptions, and is designed to answer questions of nature versus nurture, and it's not designed to predict response to selection, for example. And so this concept of reliability is quite similar to repeatability that has been used in plant and animal breeding. A difference is that repeatability can refer to non-independent measurements. For example, if you're a forage breeder and you have multiple harvests, you can use repeatability for those multiple harvests. If you have multiple measurements of different fruits on the same tree, you could look at the repeatability of those measurements on multiple fruits in the same tree. Whereas when we think of repeatability, then they're quite, the assumption is that these are independent measurements that are independent from each other. In addition, though, I think there is a very simple concept that we need to report more often. Because, for example, if I say, well, reliability of corn yield was 0.40. Yes, that gives me some idea, 40% signal versus noise. But it would even be more useful if I would report the least significant difference. For example, if I say that repeatability of corn grain yield was 0.4, and the least significant difference at the 5% level was 10 bushels per acre, then in my mind, I have this additional picture of how much of a difference do I need to have differences that are declared as significantly different from the other. All right, another borrowed concept would be just more simulations. Simulation is not new in plant breeding. Simulations have been used, and I think we need more simulations. I'm reminded of this SNL skit called More Cowbell, more simulations. We need more simulations in plant breeding. And so there's different simulation packages that are available, for example, QGene or AlphaSim. And I think where we need to go in simulations is that the entries that we have in a simulation program are not generic. First of all, we want to simulate the whole breeding process, not just the selection process, but simulate the crosses that are made and the selection of these parents. And then we want these simulations to not be generic in that the entries, if you would, in these simulations correspond or mimic the actual individuals that are available in the breeding program, the actual pedigrees of the individuals, the marker data that these individuals have, the phenotypic data that these individuals have. And then these simulations that we have can then be used to also measure the response to selection. Because now, instead of response to selection is quite interesting because it used to, in the classical sense, be due to what happens if I take individuals, I randomate these individuals and I come up with the next generation and I measure the difference in the mean. That is my response to selection. Even response to selection these days has taken a different meaning. It's more, what if I have a population of doubled haploids, I take the top 10 doubled haploids, I evaluate these top 10 without recombining them. There's still a top 10 individual double haploids. What's the change in the mean? And that has been interpreted as the response to selection. Regardless of how we interpret it now, if we do simulations, then we can still come up with responses to selection either within a given cross or across different crosses that we might want to make. So again, what I'm envisioning then is that different simulation engines that it can begin to answer in a better way, I hope, these same questions that we used to expect of the classical machinery in quantitative genetics. The last point is BLUE. And this obviously stands for just best linear unbiased estimation and best linear and unbiased prediction. Estimation for fixed effects, prediction for random effects. And the mixed model methodology is quite useful in plant breeding because, one, it allows the use of information from relatives. Second, it allows handling of very unbalanced data. And the BLUP approach and BLUE approach as used in genetic improvement was developed in the animal breeding realm, largely by Dr. Henderson. The use of BLUP in plant breeding began as a trickle. It started when Albrecht Melchinger at Hohenheim in 1987 used the BLUP approach for shrinkage of estimates of combining ability. I used it, the blue with information from relatives, used it via GBLUP in 1994. So it started as a trickle, but I'm so pleased that in not too long, there was a time when a plant breeding student would not know what BLUP and blue were, and now it's become commonplace. That we see a lot of literature in plant breeding and genetics that talks about prediction and blue and BLUP and mixed model methodology, and it's a wonderful development that we certainly have embraced. And it's interesting because the statistician Oscar Kempthorne back in 1988 made this observation about animal breeding. And basically he said that the whole theory of animal breeding has become mixed linear model theory. And that he said at that time that really the only meaningful use of genetics was in the use of Malekow's coefficient of coancestry to estimate the pedigree-based relatedness among the individuals. And so you use these coefficients of coancestry to come up with the additive relationship matrix. You use that in your mixed model equations. And now we don't even have to use pedigree-based coefficients of coancestry because we have molecular markers. And so with molecular markers, we do not have to have these probabilistic methods for estimating relatedness. And we don't even have to have formal estimates of heritability, because in GBLUP or RR-BLUP, then it becomes a shrinkage factor lambda that can be estimated just simply by cross-validation, for example. We could search in a grid for the appropriate value of lambda and then use that instead of our heritability. So what we're seeing and what we've seen is that quantitative genetics as applied to plant breeding has become more and more computational and has become more and more empirical. And it has become less and less grounded on classical theory, although very useful, limited in the sense that this classical theory is not able to capture the reality of what we might have in the biological world. And so as we look forward then to quantitative genetics and plant breeding, I foresee that more of this computational and empirical approach would be happening, and it becomes more important in our graduate curricula to focus more on the computational aspects that we have for the quantitative traits in plants. And where that might be leading us here, this is just one bit of speculation. And it's a bit of oxymoron, because I think what we need now is methods to predict outliers. Methods to predict outliers. And I say that in the breeding games competitions that I've had, it's interesting that many of the gains are not from gradual gains, but they are gains from outliers. So this is for a plot for yield on the x-axis and protein on the y-axis. And there's this individual here with a yellow arrow that is quite an outlier when you look at the whole cloud of data points over there. This is not due to epistasis, because it was an additive model that was simulated. And so a question going forward, I think we're thinking about, and where much progress can be made in breeding for quantitative traits in plants, is what are different approaches that we might be able to use to increase the frequency of outliers. And again, it's an oxymoron, because an outlier is one that is not fitting a model. And we're trying to say, well, we want to develop models to predict those that do not fit the model. But I think we all are in this exciting era in which we have new tools and new data of different amounts and of different types at our disposal. That's helpful for us as we continue to ponder how the science of quantitative genetics can help us improve the plants that we work with. Thanks so much for your attention. If you have any questions, I will try to answer them. You can put them on a chat. So one is breeding value for poultry and swine breeding. Yes, I've talked with some poultry breeders, and I still don't quite know exactly how that is done, but thank you for this comment that the breeding interest in breeding value that I alluded to in dairy cattle might not even apply to other animal species. There are other comments here that are a bit too detailed. It's more for a one-on-one talk with some of these folks who I think I know. Okay, what about genetic gain? I think, yes, as I indicated, one way for the genetic gain is to simply try to simulate it if we come up with these simulation tools. We should not focus on VA, VD, epistatic, but continues to be important and relevant. So I'm saying we shouldn't have to estimate heritability anymore. We should focus on reliability. We could estimate things such as predictive ability, but a lot of this, again, would be from empirical ways. One thing I didn't mention is even mating designs. Mating designs were a huge area of study back in the 1960s. And so now it seems like these mating designs that we have paid much attention to, at least in the literature, they really haven't been used that much in the recent past. And I think they have served their purpose, and we now have shifted to restricted maximum likelihood methods to estimate variances in a breeding program, or really oftentimes we don't need to estimate these variances themselves. It's the ratio of these variances we need to estimate. And oftentimes those ratios, if they're in a fixed range, they can be estimated by cross-validation. Reliability in a changing environment, yes, their estimates of reliability would differ as well. It's not just one estimate of reliability. Exotic alleles, translocations, and so forth. Can we talk about breeding value with complex populations? I think we can talk about complex populations and exotic alleles without having to look at breeding value. And I think germplasm diversity will become more and more important. As I'd like to say, there are three necessary things to happen for a successful cultivar to be released. One is you want good breeders, you want good germplasm, and you want good methods. We have good breeders all over the world. There are good methods constantly being developed, and I think what would become the limitation and that would distinguish one breeding program from another is the genetic diversity or the germplasm they have access to. And I think tools to better access in an efficient way this exotic germplasm, I think that's a very fertile ground for investigations in plant breeding. So there's a comment on breeding value. I did not mean to say that the parental value is not important. So parental value is important. In fact, choosing good parents is a key to good breeding. All I'm saying is that we select lines, we select individuals primarily based on their value themselves. And then those that we make as parents later on are those that have proven to have a high value themselves. Rather than the other way around. Okay, so it says genetic gain is widely used as a yardstick for genetic progress. If we do away with heritability, should we change the formula for genetic gain to include reliability instead of heritability? The answer is no. Reliability is not designed to estimate genetic gain. It's simply designed to tell you how much noise is there versus the genetic signal. And so you could do that then even in the case when you have non-random individuals, when you have a germplasm, an association mapping panel for which it is not appropriate to estimate heritability, you could estimate reliability in that situation. It is not appropriate to estimate genetic gain in a panel, and so you wouldn't estimate heritability either. And so what I'm proposing then is that we estimate genetic gain then using the simulation approach. I think many of the comments here, it's wonderful to have this constructive discussion. It has to do with genetic gain. I would like to think that most of the discussion on genetic gain would have to do with the design of breeding programs in the long term. I was a breeder with a private company for 9 years, and to my knowledge, nobody tried to predict genetic gain in any of the breeding populations. Yes, we knew it in the back of our heads, and we were interested in estimating genetic gain post hoc after the fact, but nobody was really trying to predict genetic gain. And so therefore, we did not estimate heritability routinely in the company. We did estimate least significant difference to tell us the sizes of the significant differences. All right, I'd like to close with one question. As a plant breeding teacher, do you still devote significant time in your class to teaching different recurrent selection methods? Thank you. I did talk a little bit about recurrent selection, but not as much as I had in the past. I used to have 2 meetings, class meetings devoted to recurrent selection. I had one this year, I think in part because of the COVID-19 pandemic that forced us to not have one class. But I'm finding that, for example, discussions of mating designs that has almost gone to nearly zero. Recurrent selection, only the very basics, the concept of recurrent selection, particularly with mass selection and so forth. And for us in the realm of education, that is just something to think about, because on the one hand, we have one foot, we are in the present, but we have one foot in the past and we have one foot looking into the future. And we need to train our students to look into the future, but to do that, we need to also be looking a little bit into the past. So as an educator, it's always a question of how do I balance that? To what extent do I talk about concepts that were important and that have a significant bearing on the bulk of the theory that we talk about today, that might not be used as much anymore, but are still useful to have in the back of our minds as we look at these new approaches and new methodologies. And so, yes, I think this is something that we, each of us, and as a community, would need to be looking at and just trying to see what can we do best to educate our grad students in this area. So I have 24 new messages, and there is no way that I could accommodate them. So what I would like to do then is I would like to if I can identify who you are or search you, I'll Google you and try to find your email. I'll try to respond to these individual questions and comments as best I can. Thank you very much for your attention this morning. It's been great to spend this morning with you. Thank you again. Bye-bye.